So my name is Lisanne, and I'm a PhD candidate in the last year of my PhD, and I'm working at Utrecht University. And today I would like to tell you a bit more about stem cells and how we use them within the iPSpine project. Oh, it's stuck. Okay, it's working. So let's start from the beginning. So what is a stem cell? Well, stem cells characterized by two main things, and that is that it can actually replicate itself, so that there are more stem cells, which we call self-renewal. And it's also able to differentiate into cells of different organs. So it can, for example, become a muscle cell, a blood cell, or a nerve cell. And the amount of different cell types it can become depends on the type of stem cell that it is. So on the left side, we have the totipotent stem cells, and toti stands for whole. So these cells are present within a fertilized egg cell, and these cells can become all the cells within an embryo, but it can also become the structures that actually support the embryo. So for example, the placenta. And then if we move one step to the right, we have the pluripotent stem cells, and pluri stands for many. So these cells are present within the inner cell mass, which is illustrated by the red cells. And these cells actually become in the end the embryo. So these cells can become all the organs that are present within an embryo. And then on the right side, we have the multipotent stem cells, and multi stands for several. So these stem cells can become all different cell types within one organ. So for example, it can become different cells within the brain, but then it cannot become cells of other organs anymore. So you can imagine that in the regenerative medicine field, there's a lot of interest in working with these embryonic pluripotent stem cells, because they can be used to generate actually different types of tissues that can be used in treatments for several diseases. However, as you can imagine that we have to collect them from embryos, it also comes together with a lot of ethical issues, because this fertilized egg cell can in the end become a human life. So that's why a lot of people think it's actually unethical to use these cells in research. And that is why we were very happy with the discovery of these two men. So they won the Nobel Prize of Physiology already in 2012. And they did the discovery that you could actually change a mature cell, so a cell from any organ, and then you can reprogram it to become pluripotent again. So to explain this a bit more in detail, so you can take a specialized cell, for example, from a skin sample, and then with this set of pluripotency genes, you can actually reprogram it to be pluripotent again. So we call this induced pluripotent stem cells, because we induce them to be pluripotent. And then this stem cell can again become more stem cells, so the self-renewal step, but it can also then again become cells of different organs. So it's actually a bit like bringing a cake back to the basic ingredients, so the eggs and the flour, and then you can make cookies out of it. So why do these induced pluripotent stem cells, or in short iPS cells, play such an important role in the iPSpine project? Because it's even in the title. And that is because we are very interested in notochordal cells. So these notochordal cells are illustrated by this picture. So these are the big round bubbly cells. And these cells have actually shown to have the potential to repair the degenerated disc. So that is why they are the cornerstone of iPSpine. However, there are some challenges in working with notochordal cells. And that is because we lose them so early in life. So you can here see in the embryo, there is the notochord illustrated in blue. And in children, very young children and certain dog breeds, you can still see that there is blue present within the intervertebral disc. So that is where the notochordal cells are located. However, already before adulthood has been reached, so already before 10 years old, and also in the short-legged dogs, you see that the notochordal cells actually have disappeared. And also, when we are able to collect these cells and we put them in a culture dish in the lab, we see that they change. So they lose their characteristics and they don't behave like notochordal cells anymore. So that is why the iPSpine project took the approach to actually generate notochordal cells from these induced pluripotent stem cells. So for this, we could take a blood sample, collect the white blood cells, reprogram them into this induced pluripotent stem cells, and then differentiate them towards notochordal-like cells. However, you can imagine, because I explained that this cell can still become cells of all the organs, we have to give them the right instructions to become a notochordal cell. It's a bit like you start with the basic ingredients, but you have to follow the right recipe to get a cake out of it. So that is what we're working on within the iPSpine project. And this differentiation protocol was firstly generated by our partners in Nantes. So they started with specialized cell sets where we reprogrammed into induced pluripotent stem cells. And then they give a specific stimulus to the cells. And then they end up with a kind of intermediate cell type. And then they have to give another stimulus. And then they end up with cells that look like notochordal cells. And that is why we call them notochordal-like cells. However, there are some challenges with this protocol because in the end, they obtained a very mixed cell population. So only 5 to 30% of these cells look like notochordal cells. So this you can compare a bit to the process that you want to make only a cake, but you also end up with pancakes and cookies. And another problem is that they also were still in an embryonic state. So they didn't have all the characteristics of fully matured notochordal cells. So this you can compare a bit to an undercooked cake. So within the iPSpine project, we're working on improving this differentiation protocol. And one approach is that we look into the specialized cell type that we start the process with. Because as I explained during the reprogramming, the induced pluripotent stem cells, they should actually forget all the memory from the specialized cell. However, the recent research has shown that actually some bits and pieces can retain in the memory. So you can imagine that if we start this process with a disc cell, this can actually help the cells to become the notochordal-like cells. And to test this hypothesis, we processed this differentiation protocol with disc cells and we compared it to blood cells. Another point we're focusing on is a last step within the differentiation protocol. So we try to improve the instructions that we give to the cells to actually mature the notochordal cells. And this we can do by changing the culture conditions. So for this, you can compare it a bit to actually finding the perfect baking time and temperature. And furthermore, we can culture them together with these smart instructive biomaterials. And this is a part where other partners in the project are working on. And in the end, we want to combine these biomaterials together with the cells to actually optimize the differentiation towards the notochordal-like cells. And lastly, we could also, on genetic level, give them specific modifications so that this also improves the differentiation towards the notochordal-like cells. Another approach would be to actually purify the notochordal cell population that we have in the end. So instead of, if you want to bake a cake, you focus on improving the recipe, you could also, in the end, just get rid of the pancakes and the cookies. And this we could actually do by a technique, and this is called cell sorting. And we can do this by making use of cell surface markers. So these markers are present on the outside of the cell. And what we can do is actually label these cell markers with a fluorescent dye. And then with a very fancy machine, which makes use of a laser, we can actually sort out the cells that only have this marker. So then you end up with a population that only contains of the cells that you want. However, then there's another challenge because up until now, there's no specific marker for notochordal cells known that we could use for sorting the notochordal cells out. And therefore, also within the iPSpine project, there's a collaborative effort between our partners in Nantes, the partners in Utrecht, and the partners in Hong Kong to actually keep looking for such a surface marker that we could use for the cell sorting technique. So the ultimate goal of iPSpine would, of course, be to inject these notochordal cells, and this could then stop aging or even rejuvenate the disc. However, unfortunately, we still have quite a long way to go because, unfortunately, such a therapy is not developed overnight. However, the steps that we made within this project and are still making, they do give us a lot of new knowledge that can be used in follow-up research. And we keep working on finding the ultimate treatment for helping patients with lower back pain.